get into this from a real workflow application standpoint. As I mentioned before, Kelsey and Paul were talking about how to expedite. A lot of other questions, I'll try to kind of form my presentation part here to questions. Typically, we get an investigator's brochure. We get a whole plethora of documents of prior clinical research and peer-reviewed publications from all over the world, and maybe even now information from NVIDIA's technology on designing new molecules. So we have all that data to ingest to create a protocol. So using Floyd, we can take all that data and keep it siloed within your iteration of TrialKit and create a protocol from that. So you can include all the inclusion, exclusion criteria, etc. You can use company templates. So if you have a Lilly or Pfizer template that you use for protocol, I know Merck has a certain style also. You can put that in and say, I want to use this template. Here's the IB. Here's all the research, and you can put gigabytes of data into that to have it researched to come up with that protocol. So think of this as a you're the human in the loop, and you're using this Floyd tool throughout the process of building your trial. So then from that point, you can come up with SOPs, any templates that you need for your IRB, FDA, any other regulatory agencies that might require stuff for your meetings before you even start a trial. It will develop a full protocol, inclusion, exclusion criteria. And I'm going to go off a little bit here. I'm sure we've all seen inclusion and exclusion criteria that nobody on the planet's going to fit for, right? Somebody way upstream thought it was a good idea to make it really tight. You can bring in the synthetic data to simulate. If we ran this study in 17 locations and one of them was Dallas, could we recruit enough people to meet this inclusion exclusion criteria? And Floyd will go in and give you some analysis on the potential for you having enough people to actually fit, instead of getting to a point where you've already got IRB approval, you're running the trial and you can't find anybody, right? So to that point, then you can get that information and go back to FDA and say, hey, this is great, but we want to adjust A and B because this isn't feasible for actually recruiting patients and participants that would fit into this criteria. So that goes to the endpoints and statistical models and simulating some of that.